Hello, welcome to Module 3 of this week's lectures. Till now, we have discussed different generalized aspects of ordinary differential equation based models. We have discussed how to solve an ordinary differential equation based model using analytical method by integrating in the equations. We have discussed how to numerically solve them. Along with that, we have discussed different algorithms of numerical solutions of ODEs. We have discussed how to graphically analyze using direction field or phase plane plot if I have a system of ODE, for example. We have also discussed how to create models and simulate them using JSIM. If you have noticed, in all these models, we have discussed about population growth model. We have discussed about spread of infectious diseases. We have discussed how fish growing in a tank and then shoaled can be modeled using ODEs. All these models are actually macroscopic processes involving population, individual fish, individual human being, or microorganism, something like that. Now we will enter into another aspect of dynamics in biology, that is the molecular cellular processes. Every cell undergoes, every living cell continuously undergoes different molecular processes. For example, a cell may divide, it may go through a cell cycle, as you remember. Continuously as the cell is living, it requires new proteins, so it is transcribing and translating proteins. Those transcription and translation are again controlled by transcriptional network. Then you have cell signaling, by which an external signal comes and the cell responds. Obviously, there is metabolic processes going on. Metabolism of sugar, metabolism of lipids, metabolism of proteins, amino acids. So continuously there are many processes going on in a living cell, and all these processes are dynamic processes. And we want to create mathematical models for those dynamic processes, if you remember, and particularly we want to create ordinary differential equation based models for them. So if you open any cell biology textbook, you may find the type of figure I have shown here. The figure I have taken from Cake database. It is showing a simplified and reduced model, or rather I will say it's a graphical model of MAP kinase pathway. MAP kinase pathway is one of the pathways which controls multiple processes. For example, cell cycle, it controls metabolism inside the cell. And multiple signaling molecule like growth factor or growth hormones, they give signals, input signal to this MAP kinase pathway. And as you can see in this figure, this pathway, even when we are made it a reduced form, involve multiple molecules. So if you open your biochemistry or cell biology book, you will find many such cell signaling pathways. You will find large chart or maps for or graphical model for metabolisms or metabolic circuits. And we want to create models for them, and I want to understand the dynamics of the processes involved in it. Now, there are certain issues when you are trying to model this type of cellular processes. Let us look into those issues, and some of the issues are actually will cause a difficulty in creating mathematical models. The first important issue in this type of molecular cellular processes is that these processes involve large number of molecules, in hundreds sometime. So if you have large number of molecule, and if you want to consider each of these molecule as their concentration as a dependent variable, those are changing with time, then you have so many ordinary differential equation. If I have 3 molecules, then I will have 3 ordinary differential equations. If I have 4, I will have 4 ordinary differential equations representing rate of change of concentration of each of these molecules. If I have hundreds of them, then I have hundreds of ordinary differential equations for each of them. And that's obviously a trouble when you want to simulate them. Then, apart from molecules, there are many types of processes involved in a particular network. For example, the network I have shown here, initially if you notice, this is the cell membrane, and you have here receptor-ligand interaction, Then what the processes going on this part are essentially cascades of kinase enzymes. So in phosphokinase pathways, where one molecules get activated and it becomes a kinase enzyme, it goes and phosphorylates another molecule, that molecules become phosphorylated again, and it is an active kinase now. So you have a cascade of events happening. So this is a phosphokinase phosphokinase cascades, if I say it simply. Then in this part, they are showing how transcription factors are working so that transcription are happening, followed by translation. So if you look at simply in this reduced model of MAP kinase pathway, there are multiple processes. Some are ligand receptor binding, some are phosphokinase cascade of reaction, one after another, and then at the end there is transcription and translation. Apart from there are obviously degradation of proteins using a particular pathway. So a molecular network which eventually may control suppose cell cycle or growth of the cell is made up of multiple molecules involved in multiple processes. So when you are doing creating a mathematical model, you have to take consideration of all these processes. So all these processes should be reflected in your model. One third problem that you will always face while working with large network of molecular processes is that dearth of experimental data. Till now, the type of experimental system that we have, it is very difficult to collect dynamical data. Means I want to collect the change in concentration of each molecule with respect to time for all the molecules in this network. So it is very difficult to collect information on dynamics of all molecules by experiments, and that's very rare actually. Then another important problem and rather a big obstacle in creating ordinary differential equation based model for this type of large network is that you require kinetic parameters. If you remember, if I write a simple ordinary differential equation like dx dt is equal to k into x. So x is the dependent variable, k is the constant which is parameter. So I require numerical value for these parameters if I have to model and understand the dynamics of x. So similarly, in these large biomolecular network, I need to know the parameter value for each of those parameters involved in the processes, like different rate constants. And that is very difficult to calculate experimentally, and for most of the cases, it is actually not known. So there is a dearth of quantitative information that I can plug in my model. And that's a bigger obstacle for us when we want to create a model for an ordinary differential equation based model for a large molecular network. Now, if I have these troubles, if I know these are the issues there, can I circumvent them and try to break down the problem in a simpler fashion? And that's what someone working in electronics or electrical engineering will do. Suppose I have opened a radio and I can see a electronic circuit board with lots of things attached there. So if you look into the electronic circuit of a radio, it's a large circuit involving multiple things. But you can jot down few things, you can break down this whole circuit in a small part, and each part will have different function. For example, one part of the whole circuit may be essentially a circuit for receiver, which receives signals. Then there may be one part of the circuit which is independently is nothing but an amplifier. Then there may be another part which is converting this electrical signal into mechanical motion, which gives rise to sound in the speaker. So a large electronic circuit, which as a whole is a radio, can be broken down in different parts. So can I use that concept to break down a large molecular network into small independent parts? That would help us to create a simplified version of the large thing. That means what I am saying here, let us break down a large network, the way engineers break down an electronic circuit into a smaller one. Let us break down a large network, molecular network, into small parts. So if we do that, what do we get? If I break a large molecular network into sub-part, we get sub-networks, right? And we call them network motifs. And these motifs are not just made up of one molecule. They are molecules, they involve multiple molecules and involve multiple molecular processes. So they involve few molecular processes and molecules. And one interesting thing of these motifs are that if you analyze multiple large network, for example, you take each transcriptional control network, human, a particular cell lines transcriptional network, you look into transcriptional network in E. coli, and then if you break down all these transcriptional network, which are quite large, involving thousands of molecules, and if you break them in sub-network, you will find most of these sub-networks, they are handful in numbers, but they keep on repeating. So these network motifs or sub-networks are actually repeated and connected together to create a large network. So if I can break down a large network into small repeating subunits, then what I will do? I will look into whether these subunits or network motifs has specific characteristic dynamical properties or not, or functions or not. So if I can identify certain handful network motifs which involves a handful of molecules and processes, and if each of these network motifs has a particular dynamical characteristic, and if each of them has a particular function, then I am in an advantageous position. Then I can study them separately, I can model them separately, and then I can connect them together to create a large model of the large real network. So that's what is the impetus behind looking for network motifs, and many scientists are actually working on identifying and characterizing this network motif in large networks. So let us take few examples of network motifs which are very common in biological molecular processes. The first one I have shown here is positive feedback. You look into cell signaling pathway, a large cell signaling pathway, maybe a MAP kinase pathway or insulin signaling pathway, something like that, you will see this type of positive feedback there. A positive feedback circuit has a particular motif structure. The signal comes from one molecule, this is the first one, suppose A, that activates B, B activates C, and C in turn activates A again. So the signal that is coming from top is processed by intermediate step, and then the last step goes back, feedback the information again to the initial. So there is a positive feedback. This type of architecture of network motif, positive feedback, you will find in case of transcriptional networks also. For example, A may be working as a transcription factor and controlling the expression of B. B is again a transcription factor and that controls expression of C. And C again is a transcription factor that also controls A's production. That means A will induce production of B, B will induce production of C, and C in turn will induce production of A. So it is again a positive feedback. Let us look at the second very common network motif in molecular processes, negative feedback. Just like positive feedback, it is the same thing where a feedback is there, but only the signage is changed. So what is happening? Maybe this one, first one is X, which is a transcription factor, which induces production of Y, suppose. Y is another transcription factor, and it induces production of Z. Now Z is also a transcriptional inhibitor of X. So Z goes back and inhibit transcription of X. So in a turn you can see by a delay of time X is actually represents its expression. Now not just in transcriptional circuit. Negative feedbacks are very common in metabolic circuits. They are very common in case of cell signaling. In cell signaling network, it can be like this, that suppose the external signal comes to X, that is the information coming to X, X get activated and it becomes an active kinase. So when X become activated, it work on Y and converts Y into a active enzyme. So once Y is activated, it works on Z and converts this Z from inactive form to active form. So now Z becomes an active molecule and it goes inhibit activation of X. It inhibits function of X. So I have a negative feedback. Let us look into a third network motif which is also very frequent, and we will deal with this very frequently in our modeling schemes. This is called incoherent feed forward. Let us first discuss what is feed forward. And feed-forward network motifs are very common and well known and well studied in case of metabolic networks. But of late we have also found them very regularly in case of transcriptional network studies and in case of cell signaling also. What is feed-forward? Feed-forward has a particular motif style where the input starts here at the top. Suppose this is A. Then the information passes from A to B, then B to C. But there is another path from A to C which may involve D here. So the information from A can reach C by two paths. So it is a feed forward. It is feeding the information by another route. Now these two paths can have same signage, means they may be telling to do the same thing, or they may have different signage. Let us explain that signage issue in details. For example, the graphic shown here in the motif, A is activating B, suppose, and B is activating C. I have a bifurcation here of the path. A is also activating D, but D is inhibiting C. So this path A to C is a positive path. A through B telling C that you get activated. Whereas A to D and then eventually C, this part is negative path. It is telling that no, don't get activated. So C is controlled by two paths originating from A. One is telling it to get activated, the other one is telling it to get deactivated. So it is incoherent. So that's why it is called incoherent feed forward. If I have a situation where A activates B, B activates C, whereas A again activates D and D also activates C, this is A, this I will call coherent feed forward. This type of incoherent feed forward, as I said, are you can find in any biochemistry textbook if you look into the metabolic circuits. They are very common and commonly observed in case of cell signaling pathways also, as well as in case of transcriptional networks also. Now look at into another network motif, which is very commonly shown observed in case of transcriptional network. Bifan. This is called bifan, as the shape is just like that. So what you have suppose A uses a transcription factor, maybe controlling expression of B and also Y. And you have another transcription factor X, which also control expression of B and control expression of Y. So B and Y both are controlled by A and X. So depending upon the dynamics of A and X, B and Y expression will be controlled. So what I have shown here, just few examples of very recurrently occurring network motifs, which we may observe in case of transcriptional network, in case of cell signaling network, as well as in case of metabolic networks. That does not mean that these network motifs are limited to only these four. There are many more, but all of them have this common property. They are made up of handful of molecules, maybe 3, 4, and a handful of processes, 3, 4 processes or 2 processes. They occur repeatedly in the large network. Each of these motifs has a particular dynamical properties, as well as based on the dynamical property, they may have particular type of functions. In our future discussion, we will try to model using ODEs for this type of network motif, and we will try to understand the dynamics of this type of network motifs in details. Now, as I say, these network motifs have a particular types of dynamic. What do I mean by that? Let us take an example to understand that issue of dynamics for a network motif. Let us first look into a simple linear path. What is happening here? You are giving an input signal to suppose A. A controls B, that means A is activating B. It may be an enzymatic cascade. A is an enzyme which get activated when an input signal comes, and when A is active, A work on B to make it more active, and so I get output. So suppose initially input was at input 1, a particular value, maybe 10 or something like that. And the output you have measured is actually at this a particular value, say 200 or something like that. Now if I all of a sudden increase the input to input 2, which is higher value than 1, so I am doing a step jump for the input, Then for this type of linear path, I can show by making a ODE based model or even by experiment many a time, is that the output will also increase. So output was initially like this, but then output will increase. So as the input increases, output also increases and stabilizes at a higher value. So that means input outputs are matching, changing equally. Now what is that? So this is the dynamics of a linear path. This is the dynamic characteristic of a linear path, A to B. Now let us look into the dynamic property that an incoherent feed-forward will have. That doesn't mean that all incoherent feed-forward will have the type of dynamics that I will show here, but it can have, under certain constraint, it can have this type of behavior that I will discuss. So let us look into incoherent feed-forward. I have an input which activates A, that activates B. The same input activates C also. This is the feed-forward path, but C inhibits B, and I get an output. So again, if I have kept input at input 1, a lower value, suppose I get an output 1. Now all of a sudden, if you increase input to input 2, what will happen? See, if I look into, without going into our model, if I look into the structure of the network, I can easily intuitively guess what can happen here. So as you increase input, input goes through A to B and output should increase. But at the same time, input is also going to C and it is telling B not to increase. And that, as you increase input, that path, this negative path, is also getting activated. So as I increase input, one path is telling more B to be getting activated, whereas the other path is also telling don't do, reduce the amount of B. So there must be a balance between this. Now imagine if this path is faster than this path, which is slower, then what will happen? As you jump input from input 1 to input 2, this faster path will increase the output, but after it has reached certain level, by that time this negative path will catch up and it will try to inhibit the process, so output will drop and then eventually it will settle at a lower value. So what is happening here? Actually, the system is adaptive. You have an input signal for which you have an output signal. All of a sudden, you have increased the input signal. If you have a linear path, then output will also get increased. But you don't want it. You want that the output should remain close to what was earlier. So if you have an incoherent feed-forward, then initially there will be a spike of output, and then the negative path will drag it down and it will settle at a lower steady state value. So this is typically nothing but adaptation. Imagine you require this thing in an electronic circuit. Your input current may fluctuate, so the output should not get disturbed. So you will put an incoherent feed-forward there, so that if there is all of a sudden spike of current, then there will be rise initially in the output, but then it will again fall back to the previous one. So these type of requirements are very common in case of biological cell signaling also, in case of metabolic pathways also. So incoherent feed-forward can have this particular dynamic characteristic that it can work as an adaptive motive. Hope now it is clear to you that what do you mean by dynamic characteristic of a particular network motif. And each of these network motifs, under certain constraints, under certain parameter values, will have this type of unique characteristic properties. What we have done till now, we have discussed that there are large molecular networks which control biological processes. Then we have tried to understand what if I break down them in smaller sub-network, which we call network motifs. Now network motifs can be again broken down into parts. If you remember, I said network motifs are made up of handful of molecular processes. So what are those molecular processes? I can call each of these molecular processes as elementary processes. Let us take a few examples of elementary processes. For example, ligand receptor interaction. On cell surface, you have EGFR, receptor for EGF, epidermal growth factor. So a EGF molecules comes and bind to the receptor EGFR and forms a complex EGF-EGFR. and this complex is autocatalytically activated and some other things is activated downstream. So this binding process is a ligand receptor interaction, or the binding of ligand to the receptor is actually elementary process. Then you can have an elementary process involving enzymes. For example, if you remember the old example that we have given at the very beginning, FOXO become phosphorylated by PAKT. So PAKT is the enzyme which is a kinase, activated phosphorylated AKT, and is a kinase, and it works on the substrate FOXO and phosphorylated. So this is a unit elementary process. A transcription can also be elementary process. For example, I have P15 gene which is expressed under the control of FOXO. FOXO acts as a transcription factor and it controls and activates expression of P15. So this whole thing, this whole transcriptional unit, I can consider as an elementary process. Now, a large network can be broken down in sub-network motif, and each of these network motif can be again broken down into this type of elementary processes. So if you look into the hierarchical structure, what do I see? At the lowest level, there are certain elementary processes. For example, ligand receptor binding, enzymatic reaction, production of molecules, degradation of molecule, enzymatic control of production of molecule, transcription, translation, something like that. So multiple elementary processes come together, maybe a handful of them, and create a network motif. For example, a positive feedback, incoherent feed forward, something like that. So I get a network motif. These network motifs are again connected to each other. So I have multiple network motifs, 1, 2, 3 for example I have shown here, and then all these network motifs, they are connected to each other and give rise to a large network. So if I have to create a mathematical model, I should be able first to write down ODEs for these elementary processes, because they are the building block of a large network. So what I have to do, I have to first learn how to write a simple ordinary differential equation for each of these elementary processes. Then I will club this ordinary differential equation so that I can create a mathematical model for a network motif. And then for multiple models or multiple network motifs can be connected together to create a large model for a large molecular network. So let us come to the key points that we discussed today. A large molecular network controlling a cellular process involves large number of molecules and processes. It is often difficult to study such large network as it is. It is difficult to study as it is if you have to do it experimentally as well as if you do it mathematically. Interestingly, there are recurring small subnetworks which we will call network motifs with specific dynamics and functions. So we want to break down the large network into small network motif and try to study them or create mathematical models for them. A large network can be broken down in number of network motifs. On the other hand, a network motif is made up of few elementary processes. So if you want to start modeling a large network, controlling molecular network, suppose controlling sporulation of something. So it may involve hundreds of molecules. But first break down into elementary processes and try to find out network motifs. So first create models for this network motif based on the elementary processes and then connect them together to create a large model for the whole process. That's all for today. Thank you for watching.